Okay, all right. We've only got 14 minutes. Let's get into it. So, good morning. I'm Ryan, and today I'm going to kind of go through an introduction for Memphis Meats and their patent around TED-on signaling and how they control their cell line. So we're going to go through first what is the cell line, how is it controlled, what are the signaling pathways that they use, and then in later videos we're going to go through a little bit more detail on all of those things. So let's get started. I need a pen. So, what is their patent? Why does it exist? What is the patent? So, they have a cell line. It's a GMO cell line. Right? Can you read that? I hope you can. Is it reversed? Right. And the GMO cell line is called O2K. O2K. Right? And it is porcine, meaning it's from pork. And the O2K porcine cell line is controlled by two pathways. It's controlled by two pathways. So, what do I mean by that? Well, when we want to produce cellular agriculture products, we need to do two things. We need to take our cell at the start and proliferate so that we have a whole bunch of cells. And then we need to differentiate. So that we have myofibers or whatever. Myofibers or more advanced down the muscle stem, down the muscle progression line, so that it becomes mature muscle cell, right? Because you can't eat, well, you can eat, but it's not the same. You shouldn't eat these non-differentiated cells because they are stem cells and they don't resemble, in their characteristics, differentiated muscle tissue, right? So you're going to get kind of weird cells that aren't really that appetizing to eat. So these can be satellite cells, or they can be induced pluripotent stem cells, iPSCs. So, what does this have to do with Memphis Meats and their application? Well, Memphis Meats needs a way of controlling their stem cell line in an economical way. See, normally, when we control this behavior of our cells, we use things called growth factors. Growth factors are relatively large proteins, FGF2 and TGF beta. FGF2 and TGF beta. Now, FGF2 and TGF beta help us control this differentiation approach, but they're expensive and they become ineffective over time. They're unstable. It's difficult to make them, and the way that Memphis Meats is trying to do this is produce a more economical and reliable approach to the proliferation and differentiation steps. So, let's have a look at what Memphis Meats are doing. I'm gonna go with red and purple because I'm a fan of red and purple. Right, so, like I said, the O, I'm gonna go a little bit further over to the right, I think. Yes, we're up here. So the O2K stem cell line is a porcine cell line which, when used properly, is immortal, right? So this is an O2, that's an O2K cell. Now what makes an O2K cell special? Well, the O2K cell has in its genome the KLF4 gene and the OCT4 gene. Right? KLF4 gene, OCT4 gene, important. Now, how did they do that? How did they get those genes in there? Well, they used a vector, probably viral. They don't say in the patent application, probably viral, to get those genes into the genome of that cell. Now, what those genes do is control proliferation, encourage proliferation, and indeed make the cell immortal. Because as we know, our cell lines have issues with dying a lot because cells have a finite number of replications that they can go through. So between 30 and 60 times your cells will be able to split in vivo and then they'll die, right? So we need a way of overcoming that is easy to control and allows us to differentiate when we want to, but does not cause damaging genetic variation as time goes on. That's an open issue. That's an open issue in the clean meat world. We'll get there. So that controls proliferation. But how? How do those two genes control proliferation? They cause the transcription of a number of proteins. It's quite involved and we'll go through this whole more detailed process in another video. But for now, we're just going to go through the DOX signaling cycle and how Memphis Meats is turning on these genes, right? So, we're inside our cell, we've got our DNA, right? And this signaling pathway is called a tet-on signaling. Tet-on signaling. Now what does tet-on signaling mean? It means we have an antibiotic, which is a derivative of, let me try and get this right, tetracycline, which in our case is doxycycline. And doxycycline is just in the space around the cells. It's in the media, right? Got some dox. Dox is in the media. In addition, in the media, we have RTA. And that's just in the media whenever. But we use dox as a conditional signaling thing. So what that means is we put it in when we want a particular behavior to occur. In this case, dox and RTA bind together, and the binding renders the RTA active. Active. And that means that it can bind to the DNA and cause transcription. Causes transcription. Transcription of what, Ryan? It causes transcription of KLF4, and OCT4. Cool? Right? Seems pretty straightforward, right? So we have in the media around our cells some RTA. When we want our cells to proliferate, we put in the media doxycycline. That allows the transcription of the KLF4 and OCT4 operons, which in turn causes the cell to proliferate. Now this is different to a normal cell because we've genetically modified our O2K cell line to have these two operons, to have the ability to express the KLF4 and OCT4 genes. Normal cells don't have that ability. Normal cells can't just proliferate on call when we have something like OCT4 in solution. So, What's the other approach? What's the other approach that Memphis Meats are using? What's the other approach? The other approach, which induces differentiation, is a lot easier. You don't need to use recombinant DNA techniques in order to achieve this. So what we do is we have in mixture E2, estrogen, very exciting. Now, E2 binds with myodir, right? That goes onto your DNA and causes transcription. Right? But what does it transcribe? What does it transcribe? It transcribes MyoD. Now MyoD is a natural signaling pathway that we have in a lot of mammalian cells that causes differentiation. So if we remember the diagram that I did before, it causes those proliferated cells, which we have a lot of now, hopefully, to turn into the next phase of the myoblast life cycle. So what we get is complete myofibers at the end. Myofibers. Right? So, doing too much so. We control the proliferation of our cell line using the TET-on signaling approach, which in the presence of doxycycline allows the transcription of the KLF4 and OCT4 genes. Those two genes cause the proliferation of our cell line. When we want our cell line to differentiate, we implement some E2 estrogen signal, which causes the transcription of the myodir genes, MyoD genes. And that's the overview of how Memphis Meats controls their OCT2 cell line. In another video, we're going to go through what's MyoD, what's myodir, what's OCT4, what's KLF4, how do you do the altering of this gene, how do you actually get the genes into the O2K cell line, and a little bit more detail on how the signaling sectors work, the FGF2 and TGF-beta, and why you'd want to do this as opposed to that. Okay, thanks guys.